Good morning. It's January 28th, and this is your daily brief in science. Here's everything you need to know. A new Lancet Planetary Health study warns health harms from the plastic's life cycle could more than double by 2040 if current trends persist, driven by emissions from production, use, and disposal. The analysis suggests improvements to waste collection and recycling alone would have little effect. Substantial risk reduction requires a mix of strategies, including material substitution, reuse, and reducing production of non-essential plastics. The study relies on modeling and cites data gaps, notably the exclusion of health effects during consumer use, certain chemicals, microplastics, and nanoplastics due to limited data and transparency in plastic composition. Inputs came from multiple agencies, with lead authors Megan Deeney of the London School of Hygiene and Tropical Medicine and Zhao Yuyun of the University of Exeter, collaborating with the University of Toulouse. Funding was provided by Imana, and the research appears in the 2026 issue of the Lancet Planetary Health. Even under optimistic waste management scenarios, more disability-adjusted life years are expected to accrue, though totals are lower than in the baseline. Experts call for improved waste management, better data, and updated models beyond the 2016 framework used in the analysis. Additional health harms, reported as less than 1%, relate to reduced water availability, ozone layer impacts, and increased ionizing radiation. The remaining harms also fall under this small percentage. Authors caution the estimate likely undercounts total health impacts by excluding other damage pathways and longer-term exposure routes. Lead author Megan Deeney notes this is a significant underestimation and points to the bottle life cycle, from oil and gas extraction to pet manufacture, distribution, use, and waste, as an illustrative example. A new look at melanoma treatment points to a promising direction, combining immune checkpoint inhibitors with anti-angiogenic drugs. This approach aims to enhance anti-tumor activity by remodeling the tumor vasculature and shifting immune cell dynamics in the tumor microenvironment. The mini-review in Frontiers in Immunology outlines how the combination affects checkpoints and angiogenic factors like VEGF and ANG-2, promoting vascular normalization and greater T-cell infiltration. Preclinical data suggests this synergy can reverse tumor immunosuppression, while clinical evidence indicates improved outcomes versus IC1 alone across melanoma subtypes, including advanced mucosal, acral, and brain metastatic disease. However, the strategy carries risks of cardiovascular and dermatologic adverse events, underscoring the need for careful monitoring and multidisciplinary supportive care. Future directions emphasize biomarker discovery to improve patient selection and the development of precision, individualized strategies to maximize survival benefits. CytoTape is a genetically encoded protein tape recorder that can continuously record gene regulation dynamics with single-cell spatial and temporal resolution for up to 3 weeks in living cells, and has been extended to CytoTape Vivo for weeks-long, spatio-temporally resolved recording in living mouse brains across thousands of neurons per animal. The system uses computational modeling to predict robust intracellular assembly with minimal perturbation and features a modular design that can expand to additional regulatory markers. In mouse brains, CytoTape Vivo chronicles promoter-driven gene expression histories across thousands of neurons over weeks, enabling a scalable platform for in vivo recording and tissue-wide analysis. Analyses show that divergent transcriptional trajectories relate to transcriptional history and signal integration, with immediate early genes displaying complex temporal correlations within single cells. Demonstrations in mouse neurons, glia, and human-derived cell lines reveal previously unseen temporal modes of regulatory pathways linked to cell plasticity, suggesting potential applications in distinguishing healthy from diseased brain processes and guiding future therapeutic interventions. The study, published in Nature in 2026, previews subscription content and provides supplementary materials detailing development, experimental protocols, datasets, and videos illustrating tissue-wide segmentation and signal extraction from CytoTape Vivo experiments. Future directions include integrating CytoTape data with single-cell RNA sequencing and spatial transcriptomics to create multidimensional maps of gene regulatory landscapes across cells and tissues, with potential applications in drug discovery, synthetic biology, and precision medicine by mapping how cells integrate signals over time and guiding feedback-driven cellular programming. A new study using NASA's Perseverance data reinterprets Jezero Crater as a longer, more water-rich landscape than previously thought. Researchers from Imperial College London identify wave-formed beaches and rock alteration in Jezero's margin unit that point to an ancient shoreline and extended habitable conditions. The margin unit shows signs of extensive underground rock-water interactions and contains definitive traces of an ancient shoreline, pushing back when Jezero could have supported life-friendly environments. 3 margin unit core samples and 1 from the Bright Angel Formation are planned for return to Earth under the Mars Sample Return mission for precise dating, carbonate climate analysis, and biosignature searches. The findings complement earlier work suggesting a younger perched lake at Jezero, and together describe a more intricate hydrologic history for the crater. Upstream, the Bright Angel Formation reveals thick mudstone deposits, implying a dammed lake upstream and adding to the lake and river history. In lower elevations, layered sandstones with rounded olivine and carbonate grains clearly indicate shoreline processes and a former beach within the crater. Lead author notes that shorelines are habitable environments on Earth and carbonate minerals can preserve environmental records, underscoring the importance of these Martian deposits. Perseverance observations suggest the margin unit began as igneous rock altered by carbon dioxide-rich groundwater, transforming olivine into iron and magnesium carbonates and revealing prolonged water-rock interaction. The results imply surface water persisted in Jezero with a large lake about 3.5 billion years ago, supporting past habitability. The study extends the potential habitability window at Jezero farther back in time, aligning calm, lake-bearing conditions with an ancient shoreline beneath a river delta. Published in the Journal of Geophysical Research, Planets. In 2026, the research reinforces Jezero Crater as an ideal site to investigate past habitability and the possibility that life may have emerged there. A new iron-based MOF nanoagent has been engineered to generate both hydroxyl radicals and singlet oxygen, triggering oxidative stress to kill cancer cells while sparing healthy tissue. In mouse studies, treated tumors showed total regression with long-term prevention of recurrence and no adverse effects on non-cancerous cells. Researchers are expanding testing across multiple cancer types, including aggressive pancreatic cancer, before moving to human trials. The MOF demonstrates superior catalytic efficiency compared with existing chemodynamic therapy agents and shows potent toxicity to cancer cell lines while sparing non-cancerous cells. The study is led by Ole and Olena Taratula and Chao Wang from Oregon State University and published in Advanced Functional Materials around late January 2026. This nanoagent overcomes a limitation of CDT by delivering both reactive oxygen species types with higher catalytic efficiency, potentially improving durability of tumor regression. In mouse models using human breast cancer cells, systemic administration achieved tumor accumulation, robust ROS generation, complete tumor eradication, and no systemic toxicity. The approach combines dual ROS generation in a single agent to enhance oxidative stress on cancer cells. If confirmed across cancers, the therapy could broaden CDT's reach and reduce tumor recurrence risk. Details emphasize the balance between strong anti-tumor activity and preservation of healthy tissue in preclinical results. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.